Right. Good afternoon. It's 12 o'clock on Wednesday, so it's time for the Iowa Learning Farms Conservation Webinar. I'm Matt Helmers at Iowa State University, professor in Ag and Biosystems Engineering and director of the Iowa Nutrient Research Center and faculty advisor to the Iowa Learning Farms. And as I always say, it's my good fortune to get to moderate the webinars many weeks, and certainly definitely this week have an important topic of moving towards a better understanding of bacterial impairments at public beaches in Iowa. Some of us were fortunate a few months ago, I think, to get kind of a preview of this information, and I think it's really important. And so we have Jason Palmer and Claire Hruby from the Iowa Department of Natural Resources here to talk about the work that they've done. As always, if you have questions, feel free to type those in the chat box, and we will get those to Claire and Jason at the end of their talk. So with that, the virtual floor is all yours.
All right, thanks, Matt. I'm going to be talking today a little bit about a long-running project that we've had going, kind of investigating what we know about bacteria impairments and bacteria, present at the beaches, at our swimming beaches here in the state of Iowa. Kind of our own title slide here depicts a lot of the partners that we've had over the years. This has been a long-running project, and we've gotten funding and also intellectual guidance and help from a lot of different organizations. So we don't want to forget about them as we go through this. So it's kind of good in this case to start at the very beginning, a little bit about what the underlying issue that we're concerned with when we think about bacteria impairments and folks recreating at our public beaches. And that underlying issue is the risk of getting getting sick, getting an intestinal infection from any various number of sources that could be present in the environment. A lot of these things, these pathogens that can impact you may include things like pathogens, infectious pathogenic bacteria, viruses, protozoan, things like that can be present in the environment. A lot of these things that are present in the environment that we recreate in are really tough to trace or to track to their source because they can take a lot of time to show up or manifest in a person. A person might become sick, 3, 4, 5, 6 hours or a week or more later after they interact with the environment, or after they ingest food that might contain some of these same pathogens that we're concerned with. And so because of some of the difficulty around tracking these, we have very little information about the direct overlap between these types of infections that happen in these recreational environments and the actual presence or occurrence of an illness, in a participant engaging in those activities in the state of Iowa. Another issue with tracking these viruses and pathogens directly historically has been that they're very difficult to track in real time or in a fast way in the environment in a way that we could respond to them. And so because of that, many years ago, there was an effort to look at what might be a good indicator organism that could be used to sort of use as a surrogate for comparing when we might expect to see these viral pathogens occur in the environment and affect people's safe recreation in that environment. And so over the years, and from some research done largely in more urbanized centers around the Great Lakes and in places where there are active sewage or human waste potentially reaching the environment, they found that fecal bacteria such as E. coli would work as an indicator of the overlapping conditions of concern. As you can see here, the fecal indicator E. coli is part of sort of the total coliform, the fecal coliform, and then E. coli is the smaller subset of that. Not all E. coli are pathogenic, as in this case, many are not. There's a lot of issues with folks kind of misunderstanding how E. coli interacts with humans when they ingest it. Some are completely non-infectious, and then there's a smaller subset that actually do directly make you ill. When this was first chosen, the broad understanding was that this E. coli could only remanufacture itself, could only replicate in the gut of a warm-blooded animal. And as time has moved forward and more research has come out about E. coli, there's actually been a little bit of information showing that E. coli may actually be able to replicate or at least persist for very large lengths of time outside of that gut environment, which creates a little bit of complication in using it in the way that it was initially intended. E. coli are a rod-shaped bacteria, and as I said, they grow well in the gut of a warm-blooded animal, but some can be adapted to grow or persist for long periods of time in organic or moist environments. In the state of Iowa, we lean towards using the most probable number method of measuring, but you can also use the coliforming units method. The difference between the two is really just in the lab. When we look at coliforming, they'll plate on the old Petri dish, count the number of colonies that are there and use that as an estimate. Whereas the most probable number is used by plating them in a gel pack system where they're counting the cells and seeing how many cells fluoresce and then giving us that number. This is a method that's used by our state hygienic laboratory, and most of the sampling that's done across the state uses and reports in this most probable number method. So directly in the way that we use those collection or the way that we collect those data, in the case for this project, we were looking mostly at the ambient beach monitoring program and how the data that we've collected from that program over the years has given us information about the perceived health of these environments or the perceived safety of these environments. Currently, we're sampling about 40 beaches. This ebbs and flows a little bit over time, depending on whether county or city beaches participate in the program. The program seeks to sample in the early part of the week so that they can get samples out and post advisories at these beaches by the end of the week, with the goal of giving people a snapshot of what the conditions were like, leading into that most frequent recreation time period, being the weekend period of each week. The data that are collected from these sites are done so by compositing samples, 9 points from within that recreational zone where people are interacting, and then reporting those out each week and looking at whether or not they exceed our long-running geometric mean standard of 126 MPN, or seeing if they had a high spike, so an instantaneous spike above 235. The data from this network is used both to advise people in terms of the conditions that might be present in the swimming environment, but also to see whether or not over time, whether or not individual beaches are showing a tendency to have elevated levels, from week to week and year to year. And when they do, we list that particular segment as being impaired. Currently, there's about 20 impairments from this network of sampling. And it is from that network of sampling and from that identification of a problem that we oftentimes are trying to implement or go out and work in these watersheds historically to try to figure out what's going on and what can we do about it. So going back about 10, 15 years ago, the one thing that we would do within our programs here at the Department of Natural Resources is to try to assess where all of the different problems or sources of pollutants might be coming from in the whole watershed. And so in this case, we'd be looking at E. coli. We look at the entire watershed of Big Creek Lake and start to try to do things like fence cattle out or change the methodology behind manure applications and things like that, with the intent of trying to reduce E. coli that's being delivered into the lake system. And as we were doing that and spending a lot of time and assessment and money, we were noticing that our samples that we were getting from throughout the system while we were doing this project indicated that there really wasn't much in the way of E. coli showing up outside of the area around the beach. And so that led us to really think about whether or not we're being efficient in the way that we deliver our practices and the way that we assess and look at these systems. At the same time that this project was going on, there was some literature review done to look at what was kind of happening in these lake, beach systems around the country. And there was a fair amount of evidence out there that showed that there was a disconnect globally in the timing when E. coli would show up at these beaches relative to other perceived sources. And as well as there being a lot of information about how the beach sands themselves might be a major player in how these systems manifest their bacteria concentrations in the water. And so that led us to really think about how we've been implementing our Clean Water Act programs with these non-point source pollution goals of trying to improve the safety or improve the condition of the beaches. And so we set out to design a plan to do some broad-based monitoring of a few different sites around the state to determine whether or not these conditions at Big Creek that we saw hints of were happening elsewhere. And so when we set this study design up, we were looking at whether or not when we would sample in the system, whether or not we'd see gradients in condition, in the beach sands themselves in the terrestrial zone, as well as in the water, associated with the shoreline, whether or not there were associations between what was happening in the beach sand and the water, and whether or not there were associations or disassociations with the swimming area of the beach and the kind of open lake area associated with these systems. And then we selected some sampling locations across the state that had a mix of natural lake up in northern Iowa and some, more, I guess, classic impounded systems like we have throughout much of the rest of the state. And then we did some samples from the terrestrial zone, collecting sand cores in the upper 10 centimeters or roughly 4 inches of the column radiating from the shoreline, up further up onto land. We did the same thing in the water, taking samples along the transects that kind of mimicked what the beach program uses in their assessment protocols, so that we could start to look at discrete points radiating from the beach out into the open lake to look at how that performed. And we also set up sampling locations, around the watershed where we might have wash off from the watershed, local park areas, or, areas where we've seen activity with waterfowl loafing and things like that. And in addition, we set up an alternate transect that used the same collection methodology that we used in the water at the beach, on an alternate shoreline, someplace that didn't have a beach and was more kind of reflective of the other shoreline areas of the lake itself. Jumping real quickly, just right into the sampling results we had from that 2-year period, we saw remarkably consistent results across the board at the 3 different systems that we were sampling at. We very consistently showed that the highest bacteria concentrations that we could observe were right there at the shoreline. These samples are taken right at the ankle deep zone of the beach. So just first step into the water is where most of the bacteria was showing up. And when you looked at the comparison between what we saw in that beach zone and what we saw radiating out into the open lake beyond the beach rope, we saw significant declines in bacteria out in that open lake. And when we hone in on times where we had where we would have been posting an advisory, so we had a occurrence of a 235 or higher, just looking at those timeframes, we saw even more pronounced differences between what we saw in the swimming area versus what we saw in the open lake region of this system. and then zooming out even further and looking at the system that we sampled as a whole and incorporating those alternate transects as well as the discrete points that we looked at on the individual sampling day, we saw, I chose McIntosh Woods to depict kind of the story, if you will, but all the other systems that we sampled from during this project kind of had similar results to this. We showed that on average over the whole course of the sampling, the 2-year sampling, that we had, pretty low levels of bacteria occurring in the areas that would feed from the watershed, where you move from the main bay, the bay out into the main lake, this being Clear Lake. We had, averaged below the detection limit. But you can see here that over that whole course of time, we recorded values in excess of 1200 MPN in that swimming zone. So we had consistently high bacteria concentrations in the beach and consistently low bacteria concentrations everywhere else. So to show that it's not just because of the averages forcing this, even when you looked at an individual time, so this being the time where we had the highest bacteria concentration we observed over that 2-year period from any project location that we sampled, we showed that we had over 10,000 MPN E. coli concentrations in that swimming zone. And then just when you move out into those lake transects, the average dropped down to 26. And then looking at, we had no detectable bacteria coming from the watershed, no detectable bacteria moving in and out of the bay zone of the open lake into the bay. And we had, even more interestingly, along this alternate transect right here, when you looked at the concentrations along that transect, it averaged below detection limit. So we, even looking at a gradient of a shoreline in the same way somewhere other than the beach, showed us that we had very low levels of bacteria, if any, showing up in our sampling. So this highlights that there's really something going on in this beach environment that's of concern when talking about E. coli concentrations. And so when you marry the terrestrial zone sampling with the aquatic zone sampling and start to look at the spatial trends and the concentrations over time, what we see is that we have our highest bacteria concentrations in that terrestrial beach sand area in the immediate adjacent shoreline area. So these are right at the shoreline, basically where the water meets land. This is 2.5 meters upshore, and this is 5 meters upshore. So kind of within about 15 feet of entering the lake, that sand contains the highest percentage of the bacteria that we saw in the sand. And we also showed that when we looked at on the site-specific perspective, when we looked at those increasing concentrations in the sand over the course of the year, that corresponded with increasing concentrations in the water as well. So we showed that there was an association between those higher bacteria concentrations in the sand and the higher bacteria concentrations showing up in that recreational swimming zone, but having no effect on what was happening out here in this open area. There was, consistently low concentrations out there. To get a handle on just how impactful or just the magnitude of the bacteria concentrations in this sand, these dots would have to really change here. The sand zone would just turn this whole screen red. And to show you what I'm talking about, when we start talking about the reporting concentrations of what we were detecting in the sand versus what we were detecting in the water, we have to do a very simple little conversion here of cubic centimeter of sand being equal to about a milliliter of water. When you adjust for that concentration difference, what we showed is that the sand in these situations and in these systems contained 500 to 1,300 times higher bacteria concentrations than what was present in the water on those given days, with our maximum being that 86,000 times higher concentrations on a single day, and that was actually at McIntosh Woods, the beach that I presented in the photo, or the aerial photo, just a few minutes ago. And so when you start to think about how what this sets up from a scenario perspective is that we have a very high concentration sink or source of bacteria lying in very close approximation to our recreational swimming zone. And so that recreational swimming zone is the area that we sample and we're concerned with, because this is where people are engaging in the most, vigorous activities, swimming, full head immersion, that kind of stuff. So we're really concerned about making sure that this region is safe. And the tool that we've had available to us is looking at E. coli concentrations. And so if you start to think about this high concentration area, delivering that material, whether it be through precipitation, runoff, water pushing through that high concentration area and moving it out into that recreational zone. Can also move on, relatively dry days where you have frequent onshore winds that might re-entrain material in this zone and bring it back out into the lake. And kind of, and then in this case, when that wind is moving inshore, it doesn't allow it to mix out into the open lake, kind of concentrates it in that zone. And it can also pick up other things like fecal matter that's on the beach here and move it out into the open swimming area. And then we also have direct deposition of material from waterfowl loafing or potentially dogs that people bring to the beach and things like that create complications in looking at associations between condition and when the bacteria are going to show up at their highest concentrations. And so this was the spatial component of the work that we were doing. And trying to look at these associations between how bacteria were acting or where they were showing up within a system, and leaning on that information to really guide how we thought about management of these systems in terms of what to do about the bacteria. Once we know where they're coming from, what do we do next? But at the same time that effort was going on, Claire and I started to communicate about some other more robust and assessment tools or technologies that we would be able to use to look at the actual presence of viruses and pathogenic bacteria and things like that might actually cause directly be the culprit of causing human health concerns in these systems. And so we started working with the Laboratory for the Infectious Disease and the Environment up in Wisconsin on a modified collection methodology that Claire had previously used to test groundwater samples to look at the presence of viruses in drinking water wells around the state, where we could filter material through a pre-filter to kind of get those bigger chunks of algae and other things like that that are in the water, and then move it through what's a medical grade filter. It's actually a filter that's used in kidney dialysis to remove as many of the particles, the DNA snippets from this water, and then send it to the lab where they could then run analysis to look for these entities in the water bodies. We did this on the lakes, the same lakes that we were doing the bacteria project on, and we started during our second year of assessment of the spatial component. We sampled for 17 different genetic sequences. Some of them were unique to humans specifically, others to animals. And one kind of interesting one that was a plant pathogen, the pepper mild mottle virus, which the literature is a little noisy on. It might be a good indicator of human waste, but it could also be a reflection of waterfowl or shorebird waste too, because the virus is actually unique to peppers, and so it survives or holds up very well in the digestive tract of us or other animals, and we excrete it when we urinate or whatever. And so it could be an indicator of that us eating hot sauces, ketchups, things like that, and then it showing up in these samples. But anyway, one of the things that we did actually collect that was specific to human was we had two different viruses, the adenovirus and the norovirus. Both of them showed up at several different sampling locations, but in fairly, well, I'll get to the next slide here in a second. You can see our detections here that showed that we were in that one robust year of sampling that we did at the Big Creek State Park, Hickory Grove State Park, and McIntosh Woods at Clear Lake State Park. We went to these systems in conjunction with that bacteria sampling that we were doing from April until the mid to late October, sampled every 2 weeks. And during that collection time frame, we showed at Hickory Grove that we had 2 different times where we picked up the adenovirus, which is the human virus. And at McIntosh Woods, we got kind of a flurry of different hits of the pepper mild mottle virus, 2 different human-specific viruses, and then also the avian influenza virus, which is the same thing that's responsible for some of the poultry facility outbreaks over time. The noise that's in with this 1 year of sampling that was kind of done as a snapshot of what's possible from a collection technique perspective and from a broad screening of the environment was that we saw very little consistency in whether or not the presence of viral material overlapped well with our bacteria information. The summary below the table here kind of in a confusing way essentially says that you can have bacteria present and you can have viral material present, but you can absolutely have viral material present when there are no bacteria present as well. So it brings into question whether or not that the E. coli itself is a very powerful screening tool for those conditions where you might get viral material present in the environment. So what we've started to do from the information that we've gathered from both the spatial component of the E. coli itself and the viral sampling and the partnerships that we've built with the Laboratory for the Infectious Disease in the Environment up in Wisconsin, is that first from the spatial component, what we started to do is build system-specific TMDLs where we look at the beach capture shed, the zone of influence to that beach zone. We look at bacteria concentrations or sources within that zone. We document that and model what the reductions would need to be and develop a TMDL or a reduction plan for those systems. And then we're partnering with our 319 funding. We're trying to partner with and roll out grants to state beach and county beach, city beach entities to try to put in management practices and infrastructure that can kind of limit the pathways of delivery of that material that's in that capture zone to the recreational swimming zone, as well as try to manage or reduce the concentrations of bacteria that might build up or be present in those sand and adjacent grassy areas to the beach. And so that's sort of the active component of what we're doing from an implementation standpoint or trying to get going from this. But in addition to that, we're moving towards trying to utilize some of the more advanced tools, source tracking and the viral presence and concentration collections that we kind of did for that 1 year. We're moving towards over the next 2 years, expanding those efforts that we were first playing with a few years back to expand it to several different systems so that we can look at whether or not some of these trends that we saw hold up over a range of different years and so for different hydrologic cycles and different years as well different system types. And we're also going to expand that sampling into the sand so that we get a better understanding of what the pathogentic risk of that sand is as well, because we showed from our previous spatial studies that was obviously a very important component of what was driving the bacteria concentrations on these systems. And with that, with those data collections, we're also going to be partnering with the Wisconsin lab to develop some risk assessments for the system so that we can look at from our screening what the concentrations and frequency of occurrence of different pathogens might be in these systems, and what that means from a risk perspective. So taking it kind of a step further than just looking at simply looking at concentrations of bacteria as a screening for when there might be a problem, to actually screening for the actual problem that's at play in these systems. So that was kind of a quick rundown. There was a lot more than 25 minutes worth of material to go through there, and I took a lot of slides out, so hopefully we can have a little bit of conversation and Q&A coming up here.
Yep. Thanks, Jason. So if you need a CCA, CEU, please email Alena Whitaker, so ALENAW @iastate.edu by today at 5 o'clock with your name, the name you entered to watch the webinar, and your CCA number. So Alena W @iastate.edu. As we go to the next slide, just put it out there. If you have questions, please type those in the chat box and we'll get them to Jason and Claire at the end. And next week, I'm bummed I'm going to miss it. Dr. John McMain will be talking about building South Dakota's roadmap to water resilience. So tune in next week for John. Okay, we've got a couple questions, and I'm going to ask the first one when I was wondering about. Jason, can you tell us a little about what management practices some of these beaches are looking at?
Yep. So from a couple of different things, both looking at from an active management perspective and sort of a passive management perspective, sort of like stormwater infrastructure versus what staff might do on a day-to-day basis. We've been looking at sort of implementing more things that you would think of being urban stormwater techniques, bioswales, retention cells, rain gardens, things like that, to reduce the runoff from the adjacent landscape through that zone that can accumulate or attenuate bacteria. A lot of our beaches, especially the ones that are in impounded systems or dammed up lakes, have very steep shorelines leading down to the beach, and so you can get a tremendous amount of runoff coming off a very small section of land in those settings. And then we're also working with park managers, specifically at Big Creek and then also up at the Clear Lake area, to utilize harvesters, machines that harvest material, trying to remove fecal matter as it gets deposited and other foreign things like that into the beach, out of there, and then take it off site. So those are two examples, I guess. So that removal of the fecal matter, have you tested how well the machines work? There's been some direct study on those on certain systems. In our case, we were just kind of looking at whether or not they would pick material up and remove it rather than crush it. So we used a Cheeto analogy, where we threw Cheetos onto the surface of the beach, and we ran two different commercially available collection rakes across the surface. And it was very obvious which one worked well, because on one of them we had a yellow smear across the surface of the sand, which you can imagine, if that was not a Cheeto, what that would be. And then the other one left nothing. And there were nice individual intact Cheetos inside the hopper. So that was the one we went with. But in terms of testing the sand, we have been collecting data at the Big Creek system for the last 4 years, looking at the implementation using that rake on that system on a weekly basis, over time. And we've shown that there's been a slow decline each year in the concentrations in the sand. So we have been seeing a reduction in the, kind of how hot the sand is, over time. Okay. All right, we've had some other questions come in. So the first one is, when the sand water samples were taken, were fecal deposits avoided so that that didn't unduly influence the E. coli count? So not Not super actively. What we did, we took fecal samples as well and had those run separately to just look at in different ways. But from the sand collection perspective, we took photos of, we put a 1 meter circle, PVC circle, just for a plot out, and then took photos of each of our collection points so that we could kind of look back at what, how much manure there was within a certain circumference of each collection point. If I go up to the spot to take a sample and there's, an obvious chunk of manure sitting there, we would avoid pressing the sample collecting device through that piece of manure. But after a rain event, different things like that, these pieces of fecal matter will break apart and kind of spread across the surface relatively evenly. And in those cases, no, there's really no avoiding them.
Okay.
That might be TM.
Okay, thanks, Jason. Next one was it kind of highlighted what was being done at Hickory Grove, maybe for some management, that they haze and hunt geese and conduct goose hunts in the fall to reduce the goose numbers and use of the beach there. Okay, next one is, can you elaborate how these findings may impact our overall lake watershed management practices?
Well, so this is really looking at the E. coli, right? Like just specifically in this case, the E. coli. And there are plenty of reasons to engage in very active and manure management, livestock management, you name it, to go far and away beyond whether or not those are significant sources of E. coli in the case of a recreational swimming beach. With the nutrient components and other things like that, there's very little reason to move away from that type of management. This is really good information to think about when we have this little component, right? You think about the beach shed and the water that people are engaging in these activities in. We have a sort of a critical access point, right? Like we have a location that people utilize. We invite them in, and it's kept there, for the people of Iowa to enjoy. And so when we start to think about building a plan for how best to manage, specific issues that may come up in those zones, we wanted to come up with, the best kind of information we could to drive, those management actions. But in terms of what we would do up in the watershed for a host of other reasons, I don't see this really impacting that.
Okay. Great. We may have a question. We'll see which one of you takes this. Maybe we'll get Claire involved here. But the question is, how does the health risk from pathogens in the water compare to that from pathogens in the sand?
Well, Claire, take up. We haven't sampled the sand yet, so we don't have pathogens in the sand yet. But Claire can fill in.
Yeah, we don't know, and that's a great question that we want to answer. We suspect that some percentage of the E. coli that's growing in the sand is what Jason described at the beginning, is naturalized, and not necessarily associated with an animal source. But we are also looking at microbial source tracking. We didn't have time in this talk to discuss it much, but looking at specific animal sources, and so that will inform it as well. But yeah, we want to understand. We don't want people to suddenly avoid the sand, jump over the beach and into the water. We want to understand what those risks are as well. So that will be a few years from now. We'll give that talk.
Okay. My daughter would just like to jump from the sand. She doesn't want to have sand on her feet, so she just wants to get in the water. Okay. The next question is, do you have cyanotoxin data from any of these systems?
So there are. Dan Kendall's on right now. I don't know if he has. Generally speaking, yes, we do, if that was the question.
Okay.
I will add to that folks in Michigan have been looking at correlations between the algal toxins and E. coli, and they're actually finding, algal mats as potential source of, or reservoir for E. coli. So the folks up there in some recent PhD theses are finding some correlations with the algal growth, and I'm not sure about the algal toxins themselves, but definitely the algal growth.
Okay. I guess is maybe one of you can comment on this. Are other states doing looking at similar kind of monitoring methods, topics around this area, and maybe what they're finding and actions they're taking?
So not a lot. There's been a little work kind of more out of the academic realm in Minnesota and in the Michigan, Illinois kind of Great Lakes belt, if you will. If you think about it, a lot of the money to pursue these kinds of endeavors come from funding agencies like EPA and things like that. And a lot of the money has been focused on Great Lakes and coastal beaches. We have a fairly robust beach monitoring program in Iowa, more robust than many other Midwestern states implement. And the links between the sand and the water was all kind of real time coming out in the papers and things like that as we were pursuing this effort. And so and in our work with EPA with getting some of these funding mechanisms and the team deals and like that approved, it seemed from outside of our region that there wasn't a lot of examples of that. So I can't say that I've exhaustively searched, but I feel like we're fairly unique.
Okay, great. Somebody asked Claire, maybe just to clarify, they indicated, did this person here, there's a correlation between the E. coli and algal growth, or is that just an emerging question?
Yes, there seems to be a correlation between algal growth and E. coli, which could have something to do with just the availability of nutrients. But also that algal mats could be a reservoir, have been shown to be a reservoir for E. coli in some locations. But the question is specific to toxins and E. coli, I don't think I've seen a robust research project focused on that.
Was that work done, did you say Michigan?
Yep, Michigan and Wisconsin. Again, quite a bit around the Great Lakes and the Great Lakes beaches.
Okay, great. Maybe last one in the interest of time here. Maybe you can go back, Jason, to one of the first slides. Kind of, this individual has just wanted to verify how far from shore the samples were taken. So maybe you could describe your transects and sampling points again.
Yeah, so just looking at result slide here, beach. So we got ankle. This was set up mimicking what was done at the time in the beach sampling program. So the ankle wash zone, we have a roughly knee deep point here, something around waist deep. So we're talking, 6 inches, foot to foot and a half, 2.5 feet. And then this would be chest deep, or about as high as you can get out there with your waders, essentially. So as far as you can get without being in a boat. And then this indicates the rope line. And so the rope line's a little variable. So that's different beach to beach where that rope exists. It's kind of just where the designated swimming zone is. Generally speaking, that's about 120 feet from shore is kind of the average, I would say. And then from this rope, we then radiate out 20 meters. So each of these points is 20 meters out, from the next one, subsequently out into that open lake environment to try to build that gradient of condition moving away from shore.
Great. Well, I think in the interest of time and respecting folks' time on here, I want to thank Jason and Claire for their talk today. And again, if you are on here for a CCA CEU, please email Alena W, ALENAW at iastate.edu by 5 o'clock today. And Claire or Jason, any last comments? I want to thank you very much for this webinar. As always, this will be archived, so folks can go back and watch that. I think it's really, really important, and I commend DNR for this work to understand this better on an important resource for us, the beaches.
Yeah, all I would say, thanks for having us. It's nice to share this information. I've been doing a lot more of that lately. And one of the things that comes out of that is maybe overlapping interests, overlapping concerns, or potentials for projects and things like that. And so anybody who's on here has questions that didn't get answered in the interest of time or whatever, you can reach out to myself or Claire via our emails. Fairly easy to get DNR emails. So
Claire, any?
Ditto. I would say we've heard a fair amount of folks curious about source tracking. Just beware of labs out there. They will charge a lot for that. And sample design is really important if folks are interested in looking at that. There's quite a bit of variability over time as far as contributions to the beach. And so it's just, reach out to someone with some expertise in study design if that's an angle anyone is interested in pursuing for a particular project. We're just, beginning to understand it, and hopefully a few years from now, the capability will be widespread for other folks to ask those questions.
All right. Well, thank you so much for all your time today. Thanks for excellent questions. And with that, I'm going to stop the